So welcome everybody, good afternoon. Welcome to our fourth webinar entitled The Inherited Ataxias by Paola Giunti. I'm Tanya Hermans and I'm working at the Institute of Medical Genetics at the University Tübingen in Germany. And I'm training manager for the European Reference Network for Rare Neurological Diseases. And I will be hosting this webinar today together with my colleague Annemarie Post, who will be co-host. So we are glad to open this webinar, which is part of our joint webinar program together with the European Reference Network for Neuromuscular Diseases and with the European Academy of Neurology. You're all muted at the moment. I hope you can hear me well. Anyhow, if you have any questions of for technical aspects or administrative aspects, you can ask them in the chat box, and otherwise you can questions about with medical content, you can put or write them in the box Q&A, which you can find at the left side in the lower toolbar. This webinar will take around 35 minutes, and subsequently there will be this questions and answer session about 15 minutes. Afterwards, after the end of the webinar, there will be in your browser, there will be one window opened automatically with a post-webinar survey. So it would be great if you can just enter some feedback from your side about the webinar, so that we can always work on the continuous improvement of the webinars. So target audience of our webinars are normally neurologists, neurology residents, pediatric neurologists, geneticists from R&D member hospitals or not. So we have also announced this webinar very widely. This webinar is being recorded, and along with the slides, the recorded version will be put at our website, which you can see here. And you can also find further useful information, so under the tab Disease Knowledge, also at our website, which is here, the link. Yeah, and as I said, please fill out the post-webinar survey. This will be very useful for us. Okay, so thank you, Paola, for joining today. We are very, very glad to welcome you and to have such a renowned expert today in our webinar. So I will just present Paola shortly, and then we can start with the webinar. Paola is a professorial research associate at the Department of Clinical and Movement Neurosciences at the UCL Institute of Neurology in the UK. She's lead neurologist of the London Ataxia Center, which is a center of excellence she established over 10 years ago in partnership with the patient organization Ataxia UK. And Paola's research interests are the pathogenesis of neurological disorders, in particular of the inherited ataxias and other movement disorders. She's investigating the mechanisms that underlie neuronal degeneration in the central and the peripheral nervous system, and her research is highly translational. She is an active and major contributor to 2 European consortia focused on the natural history of the inherited ataxias, EuroSCA and EFACTS. And she's also involved in the translation of basic research findings into the clinic for use in both in diagnostic testing and in therapeutic trials. She has been a PI and co-PI in multi-center, multinational drug trials in Friedreich's ataxia. And in 2016, she has received the Health Professional Award by the European Federation of Neurological Associations. So thank you, Paola, again for being speaker today and for accepting our invitation. And I will just now stop sharing my screen and you can start with your presentation. Yes.
Hi, good afternoon, everyone, and thank you for all coming, and I'm delighted to have you here. So I'm going to speak about the inherited ataxia, clinical and genetic aspect. So at the beginning, I would like to know what is your professional background?
So I will start now this poll, and you can just answer live this question so that Paula has an idea of the attendees being there today. Okay. And those are the results.
Good. Thank you so much. So you're all neurologists, the majority, and I'm going to talk also for the non-neurologists about ataxia. So just to be on the same page, I'm going to define what ataxia is. And ataxia is a Greek word. It is without, and tax is order, lack of order. The main features are the gait ataxia, when you have a broad-based gait with a loss of balance and steadiness, and irregular wide-based gaits. Limb ataxia means that the movements are irregular, fragmented, and there is tremor as well. For the eye movement is also affected, and they are interrupted, and there are some intrusion of saccades, so there is quite characteristics, and also nystagmus. And the speech is slurred with impairment of speed and volume. I'm going today to focus on the inherited ataxias. So the hereditary ataxia might be congenital, episodic, or progressive. And the way to inherit it, the ataxia, are mainly for dominant, recessive, X-linked, and mitochondrial. And the prevalence is 8.9 in 100,000. So the autosomal dominant and recessive ataxia are a very complex group of neurodegenerative disorders. And this has been first, a little bit of history here, and has been first identified with Pierre-Marie in the seminal article in 1893. And then was the late Anita Harding that tried to make order in this very disorderly chapter of neurology. And she came out with a classification that is very, I think, useful for clinicians, and dividing the group of inherited ataxia in three. The first is characterized by patients having cerebellar ataxia, but in addition to that, soon after they started, because the majority of patients start with rather pure cerebellar ataxia, but then the clinical pictures enrich in much more multisystemic condition. And in addition to the cerebellar syndrome, they are variably associated with ophthalmoplegia, pyramidal and extrapyramidal signs, seizures, dementia, and peripheral neuropathy. For the DCA type 2, the features is all the DCA type 1, but in addition, there is a typical pigmentary maculopathy. And then for the DCA type 3, there is a rather diffuse Cerebellar syndromes. Now, in the era of genetic testing, you need to bear in mind that the DCA type 1 are the most frequent. These are SCA1, 2, 3, and 17. The DCA type 2 is actually genotypically very clearly SCA7. And for the DCA type 3, especially if you are in this country, in the UK, you will see a late onset over the 50s years old age at onset, and a rather pure and very slowly progressive. And this is clearly an indication of SCA6. So still in these days, this is useful for an indication of what the patients genetically had. So the epidemiology, what I said, is a 0.9 to 3 on 100,000, depending obviously on the court. And in the same geographical area, you can have as well in some areas very much a high frequency. And this is especially, for example, when we look at Cuba or Azores Island and in Calabria, for example, the south of Italy, where respectively you will have a very high frequency of SCA2, 3, and 1. So what is the underlying mutations of the SCAs? Well, essentially I would divide it in 3. So the polyglutamine expansion, these are also SCA1, 2, 3, 6, 7, 17, and the DRPLA. And these are lying in the coding region of the genes of these different diseases. And then there is a non-coding expansion on SCA8, 10, 12, and 31, and 36. And then the conventional mutation, there is SCA5, 11, and so forth. And these were much less frequent compared to the first group. So I would like to talk you through this graph and this cartoon. And I would like you to concentrate especially on the left up corner, and you will see the CAG polyglutamine expansion. And this indeed are all expansion from the ORF. So this is the cartoon of the genes, and you can see ORF, intron, ORF, and 3' UTR and 5' UTR at the other end. And so this one here is the graph that it indicated the number of CAG repeat. The green is the range of the normal, then the orange is the one of intermediate, and the red is the one of the expansion. And even at glance, you can see that despite all these obviously conditions are due to the mutation, but also genetically there is a differences and the variability, number of CAG repeated, and the normal and pathological intermediate. So what is the common clinical genetic feature shared by the CAG repeat? So there is a variable age at onset, the majority is between the 3rd and the 4th decade, but the condition can come anytime and can start at any time, even at a few months of age or after 80 of age, 80 years old. There is a strong correlation between the number of CAG repeat and the age at onset. This is particularly true for the SCA7 and less for SCA2. So there is a large expansion leading to a much more aggressive and severe phenotype with the reduced life expectancies, whereas the lower number of CAG repeat, they indicate a much milder later age at onset and much milder progression. So there is a phenotypic variability within the separated diseases and also within the same family. And this is due to the instability of the CAG repeat, even within the same family. So other common clinical and genetic features is the anticipation with intergenerational instability, and the DLPLA and SCAR7 is the most anticipated of all the other families. They have anticipation that is extremely unstable. And the parent of origin expansions usually via paternal descent with somatic CAG instability for all this polyglutamine due to an increased number of mitotic division preceding male gametogenesis, and also the concentration for the DNA repair. There is then, for the neurological point of view, neurodegeneration. It's really selective for each disease. The other features that is in common is that when the number of repeats are very high, the age at onset, as I said, is very early, but they are much more multisystemic at the onset. Whereas if you start a condition in later on, it's more pure cerebellar ataxia, and then later on you add in many more symptoms and signs. And so this is tables that showed the small repeat, medium repeat, large repeat, and very large repeat. And the very large repeat is sometimes so dramatically different that, for example, in SCAR7, you have a predominantly cardiac presentation in children. Now, looking at the progression of the disease, this is a very important study that we did, and this was part of the EuroSCAR, looking at 536 patients and looking at the progression, and the progression was, excuse me, was then done by Sarah score, the rating scale for Ataxia, and we managed to monitor and to see the progression of the disease. And SCAR1, it's much more aggressive and rapidly evolving compared to the SCAR2, SCAR3, and the SCAR6 is then much more lenient. And there is a, as I said before, massive correlation, inverse correlation between the number of CAG repeat and the age. But as you could see, for example, patients with the same number of CAG repeat, they have sometimes more than 20 years of difference. And this is prompted us to look into this and to start studying and identify modifiers. So my postdoc here, Suran Netizinger, looked at SCAR1, SCAR3, SCAR17, and looking at the repeat configuration at the level of the DNA, it was very clear that the expansion as well has some interruption. And the interruption are the in red, CAT, for example, and CA, or the other SCA2 and 3 and 17. So the question was, what was the role of this interruption? Were they able to modulate the disease? And Suran took the lead on this and inspected 36 DNA of patients. They were very well studied in my court, and the classical correlation was present, but when we took and looked at the interruption, we understand that there was an interruption in 4 of these patients, and then the Pearson correlation was much tighter with a much higher statistical significance. And the uninterrupted, if you then take the uninterrupted long CAG repeat, they were falling into the, clearly into the interval, making the linear correlation much tighter. Conversely, when we look at the SCAR6, then the SCAR6 was with anticipation, instability, and the span repeat is much more discrete, from 20 to 33. And with direct sequencing, we could see there was no interruption, and the variability in the family maybe due to epigenetic factors. Moving forward to the SCA17, the SCA17 is autosomal dominant cerebellar ataxia that has a very, very variable phenotypes. They have patients with ataxia, but also with extrapyramidal sign, typically the Huntington disease-like type 4 according to the classification of Huntington disease-like, and they have cognitive impairment, they can have psychosis and seizures. And this is caused by the polyQ expansion in the Tata boxing. So he took 30 individual and cloned all these individual, and the age at onset was available for 21. And it found that there was no correlation when we took the uninterrupted number of CAG repeat and the age at onset. There was a correlation where we took the total number of CAG, including as well the interruption. And this, we wanted to understand if the interruption has a say, for example, in the presentation of the phenotype. And so we look at the interruption when there was dementia, ataxia, chorea, Parkinsonism, but they were all very evenly distributed. So in conclusion, the SCA1, 2, 3, 7, 17 has common clinical genetic picture. However, the SCA1 show more rapid progression, as opposite was SCA6, and the interruption on SCA1 is really the only now translational modifier that can be really be used in clinic to enrich genetic counseling. And the interruption delay, the age at onset, and the longest uninterrupted CAG should be considered rather than the total repeat length when we have, for example, predictive tests and genetic counseling. And the repeat instability can occur even in the presence of the CAG interruption. So despite sharing some clinical and genetic aspect, we more and more looking into this group of this condition, we see as well differences that are relevant and specific for the disease. The SCA1, the interruption, the SCA6 have no interruption, for example, and the SCA17 has interruption present in expanded alleles. So the interruption are not modifiers, basically. And no particular repeat configuration is associated as well with the phenotype. So moving forward now with the recessive condition, I'm now going to talk about Friedreich ataxia. Friedreich ataxia was identified by and described by Nicholas Friedreich in his seminal work in 1863. And this is a recessive condition, and it's the most frequent conditions, and the differential diagnosis is broad. And we are going now to talk later on, after I describe Friedreich, about ARSACS and SPG7. That seems to be the most frequent compared to all the others. So Friedreich ataxia has an inheritance, it's autosomal recessive, and usually the onset is less than 25 years of age. The prevalence is 1 in 50,000, and the carrier status is 1 in 70, 1 in 90, depending on the cohort studied. And the clinical features are characteristics with a sensory, and later on sensory cerebellar ataxia, and the dysarthria, pyramidal weakness, Babinski signs, and the sensory loss. In addition, there is as well hearing loss, the optic atrophy, and distal amyotrophy. What is really interesting for the Friedreich is a really truly multisystemic, that not only with a neurological system, but also with extra neurological manifestation. So there are kyphoscoliosis, pescavus in patients. They are very frequent. Hypertrophic dilated cardiomyopathy, there is a 70% of the patients. and it is the most frequent cause of death. So it's really relevant for, in the progression of the disease. And then diabetes and glucose intolerance is 10 or 30%. Like for the pathology point of view, the target is the DRG, large neurons degeneration, and together with spinocerebellar and pyramidal cuneate gracile tract, we're relatively sparing of the cerebellum. Recently, we have as well done a study, Friedreich ataxia and urinary bowel and sexual symptoms. And this is very important. I would like to highlight this one to the neurologists, because patients really do not volunteer to talk to you about bladder, sexual, or urinary symptoms. But these are very important, very relevant for the quality of life, and we can have some treatment for that. So in addition to the classical phenotype, the Friedreich ataxia has a typical phenotype, and we have the Friedreich ataxia with retained tender reflexes, the late onset, Friedreich ataxia with a pure spastic phenotype. Some of them has very rarely dystonic features or chorea, and there is as well the pure neuropathic variants that is very much difficult to distinguish with Charcot-Marie-Tooth. So this is a MRI, and as you could see, there is a relatively spare the cerebellum, but only there is the thinning of the spinal cord. The gene in the Friedreich ataxia, the mutation is a GAA repeat in the intron 1. The number of GAA repeat in control is 6 to 33, the pre-mutation 3465, and the patients are above 66. And there are many other point mutations, but these constitute only 4% of all the mutation. 96% remain. Most frequent is the GAA repeat, and in some of the patients as well, very rarely, there are as well deletion. So what is the mechanism for which we have a downregulation of frataxin, that is a protein that is relevant in Friedreich ataxia? So there is the unaffected and control, you produce your frataxin, but then when you have a GA repeat that is in a pathological range, there is a downregulation of frataxin. Richard Fesser, suffering Imperial, suggested that this is due to heterochromatization, and this is an hypothesis that is still very much followed, and one of the main studied. So the heterochromatization is due to, this is the hypothesis, to the large GA repeat, and what Richard Fesserson as well identified, that some HDAC inhibitor like nicotinamide vitamin E3 is able to revert this mechanism. And the heterochromization, we collaborate very tightly with Richard, and we published preclinical studies for which we took the blood of the patients and put it in contact with the nicotinamide, and we saw upregulation. And from there, we started immediately to have the phase 2 open label trial of nicotinamide, where we could prove that in 10 patients, and this has been published in the Lancet, there was indeed a reduction of the heterochromatization. So there is a decrease in DNA methylation, H3K9ME3, around the GA expansion, that is a hallmark of heterochromatization, and here as well the increase of histone acetylation. So the results consistent with the hypothesis that nicotinamide can reduce the heterochromatization. So, but more importantly, patients have upregulation of frataxin, and the frataxin came at the level of the carrier. So the patient that was here before this was before the some administration of nicotinamide. So the nicotinamide is rapidly absorbed by oral route and safe, well tolerated. There has been some side effect with nausea, but then has been controlled with the antiemetics, and there is a self-limiting raise of LFTs. So the NICOFA now is a reality, and this is the trial that is going to be multi-center, multinational. It's going to start hopefully in March and April. And this is different sites: UK, Germany, Austria, Italy, Spain, and France. And we are going to recruit 225 patients in total, and 90 of these patients will be recruited in the UK. These are the inclusion-exclusion criteria, so the age is between 18 and 50, and have a molecular genetic diagnosis of Friedreich ataxia. There are specific inclusion criteria, especially for the level of disability then looked at with the SARA score. But one thing that is as well very important, we have obviously seen the upstream mechanism, and we have addressed the upstream mechanism thanks to the upregulation of frataxin from nicotinamide. And we wanted to now look at the downstream effect. We know that frataxins are in sulfocaster protein inside the mitochondria, so very relevant to look at the mitochondrial health. And Rosella Beatty in my laboratory has looked at the mitochondrial bioenergetics, and she had identified the decrease of activity of complex 1, and also the increase of lipid peroxidation with a clear deregulation of membrane potentials in the mitochondrial membranes. So this one was very relevant, because then this has been assayed that then could be useful for then look at compounds that could ameliorate these features. So indeed, we have seen the OMAV. So OMAV is an NRF2, and this has been done then for thanks her work. The other pharmaceutical company could roll out this ambitious project for clinical trials, and we are one of the sites, and also the Retrotop with a DPUFA was used with our assay, and was able to ameliorate. And so we have now the MOXIE studies. The MOXIE study has been started a year ago, and we have just concluded, and the other pharmaceutical in November has come out with a very exciting press release, because the endpoints were achieved after the 48 weeks of treatment, and now they're looking to the FDA to have this product license. So in conclusion, FRDA is the most common inherited disorder. The clinical spectrum encompasses the early onset Friedreich ataxia, the late onset, and then the classical presentation. The GA expansion constitute 96% of the mutation, and 45% is a compound heterozygous. Screening of a large cohort of patients with a non-classical phenotype showed that there was 3 and 2, so only 0.15% were positive. So if you do not have the classical phenotype of Friedreich, there is no point to ask for a genetic test. clinical trial is a reality, and this has ended. We are in the extension phase. This is an exciting time, and see if this will be licensed, because if yes, this will be the first drug that could be licensed in Friedreich ataxia. Then we're starting to prepare for the NICOFA trial with the nicotinamide, and this is again a multi-center, multinational randomized clinical trials. Moving now towards ARSACS. So ARSACS is autosomal recessive spastic ataxia of Cerebellar, and this is because it has been particularly frequent in the region in Canada, where this condition was first described. And typically, the Canadian founder mutation is the C8844 deletion and the C750CT. And these two mutations are obviously a founder mutation. That is why they are very frequent. But now we have found the ARSACS all over the world. So before, it seems to be there was some private mutation, but now this is not the case. So the SAX gene is in chromosome 13q, and the macrodeletion, deletion, substitution, and also common mutation has been described so far. The mean of age at onset is 15 of age. So similar to Friedreich ataxia, there is a slowly progressive ataxia with spasticity, neuropathy, and then some foot abnormality, but not in all. There is in 20% some hearing impairment, blood, and sometimes epilepsy. What is very distinct from the Friedreich is this is a demyelinating sensory motor neuropathy. And for the MRI, we can see that you have a cerebellar atrophy, thinning of the corpus callosum, atrophy as well of the cerebrum, and thinning of the spinal cord. But then we have, especially in T2, this hyperintensity they call the tiger stripe. These are, though, not in all the patients, because in our series is only on 75%. And the other hallmark has been described first in Canada was the retinal nerve fiber layer thickening, looked at with a fundoscopy. However, also this thickening was not present in all patients, and therefore it was our interest to understand if we could find any biomarker that were much more specific and sensitive. And it was my research fellow, Dr. Michael Parkinson, that embarked himself in a very large study looking at 200 ataxia patients and screened them for the OCT. So the OCT is the optical computer tomography. It's very easy, inexpensive. It takes only 15 minutes, and we look at the thickness of the optic nerves. You can see here in that the ARSACS patients are the ones that are clearly stuck up from the rest of it. The 119 micrometers were really clearly cut off, and interestingly, the opposite is Friedreich. While ARSACS are a thickness of the optic nerve, Friedreich had the thinning of the optic nerve, and this was shared among others ataxias like the SARS and other ataxias that are more rare. So the rock plot, as you see, this test sensitive 100% and specificity of 94%. So in conclusion, the RFNL, thickening and OCT, is a sensitive and specific marker for ARSACS amongst a population of patients with suspected ataxia. We are now very lucky to have our neuro-ophthalmologist clinic that run alongside our clinic, and patients that come for us for the very first time also go to see them and track the OCT. And sometimes this is very incredibly, although the study was done in 2018, we still found patients and identified new patients through this technique and through this test. And then the proposed cutoff is 119 micrometers. However, caution with the carriers, because sometimes the carriers has a kind of intermediate thickness. The other cases of ataxia generally have normal or thin RNFL, especially the Friedreich ataxia. And the ARSACS patients rarely have visual symptoms, so this doesn't mean that something will happen on them for what is regarding visual acuity. So this thickness seems to be independent of the function. OCT should be a part of routine pre-genetic screening in patients with spastic ataxia and idiopathic. So moving now towards the SPG7, this is another important condition that comes in the differential with Friedreich ataxia. This is a most frequent from a late onset spastic ataxia, prevalence 1 in 900,000. Progressive bilateral leg weakness, spasticity is one of the main features. However, we have patients, for example, that started with rather pure cerebellar ataxia and then developed after quite a few years clear spasticity. The peripheral neuropathy is not very common. This can be a distinguished feature with ARSACS and Friedreich ataxia, because by autosomal recessive, rarely dominant, and the phenotype is indistinguishable from many other hereditary spastic ataxia. In some cases, as Pescavus, and the one who has peripheral neuropathy is an axonal peripheral neuropathy. So the gene is 49364 base pair, consists of 22 exons, chromosome 16, and encodes a mitochondrial metalloproteinase protein that is called Paraplegias. This is the very busy slides. I apologize, but this is a very good paper from Chana Manduma, and I suggest you to read it because it's one of the largest cohort of SPG7. And as you could see, the mean age at onset is 41, so definitely older compared to Friedreich ataxia. And there are, as a features, the characteristic is 98%, a little bit of steadiness, the stiffness of the legs, spastic paraparesis, increased tone, as well in the upper limbs and lower limbs. So there is a cerebellar sign, but these are quite mild. And there is sometimes only 22% you have a reduced vibration sense, with 99% of the patients have normal pinprick. And the tendon reflexes are brisk with Babinski signs. And the gait is typically spastic ataxia. So, but what is very interesting that the MRI shows intensity of the dentate nuclei. So this can be a very good clue for SPG7 genetic diagnosis. So in conclusion, mutant SPG7 is a common in sporadic ataxia, and patients with British ancestry has one C1529 CT allele represent the most frequent mutation, and the clue about the SPG7 often is given by the MRI of the brain showing in T2 a hyperintensity in the dentate nucleus. So in conclusion, I think that it is very clear that the dominant and the recessive inherited ataxias are a very complex group of disorders. And the best way to manage this is with a multidisciplinary clinic. Indeed, we have seen lots of symptoms: spasticity, tremor, dystonia, scoliosis, bladder involvement, sexual function, audiology for the hearing loss, and depression, especially, something that is always very, very well masked in patients with difficulties in the speech and hearing. So definitely a multidisciplinary approach is warranted in these cases. And the other things that I would like to highlight is that there are guidelines. These guidelines are now absolutely ready, available online on Ataxia UK, and you can download it. But also we have published these guidelines a little bit and updated as well in the Orphan Journal of Rare Diseases in 2019, last year, and also the Diagnosis and Management of Progressive Ataxia in 2019. So they're quite recent, and these both have been endorsed by the ERN-RND together with the guidelines for Friedreich ataxia. And these are also important. They have been, it's very thorough. It goes from all kind of symptoms and also from genetic cancer. So my talk finish here. I would like to thank you, but I would also like to submit to you some questions.
Okay, so I will fade in the first question. Let me.
Sorry, I cannot hear you.
Yeah,
I cannot hear you.
I will just now start the poll, and now the participants can answer the question and participate in the live voting. We can leave the poll open for 40 seconds or something like this. Okay, are there still a couple of answers? Okay. So now I will end the poll and show the results. Here are the results. Maybe you can comment on them, Paola.
Yeah, well, yes, that is good because this is the right answer. So 77% have all the right answer. So congratulations to all.
And then we can go to the third and last question about inheritance types. There it is. Okay, 10 seconds more. Here are now the results.
Perfect. Also 77% that's the right answer. Congratulations again. And thank you very much for being and stay with me until the end.
Okay, so Paola, I think we have one question in the question and answer session or box.
Yeah.
So this is it. What are the primary outcome of the FRDA new clinical trial? Have you considered?
I'm sorry, I cannot hear you.
Do you see the question on the screen?
No.
Oh, okay. So again, I will shout louder. What are the primary outcome of the FDRA new clinical trial? Have you considered using kinematics measurements?
Yeah, we have. And we are working towards implementing this in our trials. So it's a wearable technology that we have, and they have been developed for the last 5 years at Imperial. And one arm of the UK side, the one at Imperial, the 50 patients who will be enrolled in the nicotinamide will have the opportunity to use this wearable device.
Mm-hmm. Okay, I don't see who asked the question, but I hope that you're Paula answered your question. So again, which are the most common ataxias with dopamine depletion?
So I can see now. Two questions. Can I repeat the question? So, do you and your institution use drugs such as riluzole? So we did only in one patient, but then unfortunately we were not allowed anymore for legal reason here in the UK, because it's not licensed for the scars. So my experience is quite limited for that. The other anonymous attendees is which are the most common attacks with dopamine depletion. Well, we had the SCAR2 and SCAR3 that can present with Parkinsonism, but also for my experience, there are one subgroup of SCAR6 that can present with Parkinsonism or with proper Parkinson disease, because they are responding very well to dopa agonists and dopa. So both the three of them for our experience.
Okay, so there is another question which we have received in the chat. Can FRDA patients from other countries be included in nicotinamide study?
Yeah, definitely. So there are some differences, obviously, because the regulation is slightly different from country to country. But I would suggest to ask the center that is nearer of the consultant neurologist who is going to refer the patient to, and have a discussion with that center.
Okay, so Natasha, I hope that this is
Thank you very much.
Your question. Okay, then
Bye.
again.
Bye, everyone.
Paola, there is another question, if you can answer that.
Sorry?
Yes, there is one or two questions more. Do you have time to stay a little bit more?
Yeah, so one is for a gene therapy, I can see it now, be
Yeah.
an option in treatment of any specific ataxia. Well, for the ASO, the antisense oligonucleotide, there are lots of pharmaceutical companies that are working very hard on try to identify the right ASO, and we can use the example of the Huntington disease, and therefore I think that this is going to be a reality in a few years' time. Obviously, the Huntington disease is ahead of us for the spinocerebellar ataxias, but I think it will follow a very similar path. And for the valproic acid for SCA3, we haven't actually start any patient with the valproic acid, but we have quite a few patients who has valproic acid for other reason, and they are my again, my experience is limited, but I could not see any great effect. All right.
Okay, so I think there are no more questions.
I will leave you because
yes, you
time.
have maybe other obligations. So thank you everybody for
Exactly.
joining. Thank you,
Yeah.
Paola. And you can find the recorded webinar on our website, as I said previously.
Thank you.
And good afternoon.
Bye-bye.
Bye-bye.
Bye-bye, everyone.